The overall goal of this procedure is to visualize monocyte trafficking in the skull bone marrow of a living mouse using 2-photon imaging. First, the mouse is anesthetized and injected with a solution of rhodamine dextran to label the vasculature. The mouse is then placed in a custom metal support, and the scalp, from the back of the parietal bone to the frontal bone, is removed. A rubber ring is then applied directly to the skull with surgical glue, and the reservoir is filled with liquid saline. Finally, multiphoton microscopy is used to track fluorescent cell development over time within the skull bone marrow.
This method can help to answer key questions in the field of immunohematology, such as the mechanism of monocyte mobilization in steady state or after chemotherapy.
Thought this method can provide insight into the behavior of medullary monocytes, it can also be applied to study other bone marrow cell subsets, depending on the specificity of the fluorescent reporter used.
The following protocol utilizes a multiphoton microscope coupled with a titanium sapphire crystal laser and an acousto-optic modulator for laser power control. To enable simultaneous recording of 3 fluorescent channels, the configuration should include 3 external non-D-scan detectors, or NDDs, with a combination of 565 nanometers and 690 nanometers dichroic mirrors, 565, 610 nanometers, and 550 nanometer bandpass filters.